Hello everyone, my name is Saurabh and today we will be discussing about mathematics for biotechnology. A lot of time students who are pursuing courses in life sciences, maybe biotechnology or biochemical engineering, they come across problems or any courses in life sciences, they come across problems which need you to have a basic understanding about mathematics. So in this video, we will discuss about what topics and what things are necessary for a biotechnologist to understand. Um, as I told you all, I am Saurabh Gurav and I am studying bioprocess technology. I am GATE, GATB and DBT JRF qualified. So let us begin. Try to understand the first question. The first question is Saccharomyces cerevisiae produces ethanol by fermentation. The theoretical yield of ethanol from 2.5 grams of glucose is dash grams. So here we are in the question, it's been said that Saccharomyces cerevisiae produces ethanol by fermentation, and we know fermentation is a type of anaerobic respiration where the glucose is broken down into ethanol and carbon dioxide. So for this question, you will need to have understanding about the basic balanced equation for the whole reaction. The whole process can be summed up into this one reaction wherein you have starch. You can see here the equation mentioned. There is a starch molecule which is broken down to glucose, and glucose is further broken down to ethanol and carbon dioxide. So from a chemical reaction, you can understand that 1 mole of glucose is utilized to produce 2 moles of ethanol and 2 moles of carbon dioxide. So, but we need our answer mass, that is grams. The unit is grams. So from molecular weights, you can find out the molecular weight of glucose, which is 180 grams. So 180 grams of glucose will give you 92 grams of ethanol. So where these values come from, 180, 80 and 92. 180 is 180 grams. This value comes from the molecular weight of carbon, hydrogen and oxygen. So 6 into 12 plus 12 into 1 plus 6 into 16 will give you 180 grams. Similarly, you can calculate the value for ethanol, which is 12 plus 3 plus 12 plus 2 plus 12 plus 16 plus 1. And the whole value into 2, because there are 2 moles of ethanol being produced. We equate for 2.5 grams. So the answer you will get is 1.277 grams of ethanol. So this is very important to keep in mind that the unit is grams, and you can also calculate the moles directly from the equation given here. So this is the answer, 0.277 grams of ethanol. Move on to the next question. The next question is, in a genome with average GC content 40%, the expected frequency of occurrence of tetranucleotide AACG is. So this question has mentioned the average GC content is 50%. That's what the question is. You have to find out the probability or the occurrence. You can say probability or occurrence or frequency of that particular 4 nucleotide sequence, which is AAGC. Now, as we know that GC content is 40%, so 80 content will be 60%, and as G is equal to C, so both of these quantities, the G and C, will be 20%. So probability, so what we are going to do is, in order to calculate the probability or frequency of this particular nucleotide, we are going to multiply individual probabilities of one particular base seen in the tetranucleotide. That is, A is 0.3% into, again A is 0.3 into C is 0.2 and G is 0.2. So you get the answer probability, that is 0.0036. So one thing to keep in mind is that probability will be always less than 1, and you are told to calculate. Suppose if you're told here to calculate the percentage, so what you're going to do is just multiply the given probability or frequency with 100. So you will get the percent of this tetranucleotide is 0.36%. Let's move on to the next question. The next question is, in a diploid organism, the genes are A, B, and C, which are present in the same order on the chromosome. And AB is 10 map units apart and BC is 20 map units apart. And in an heterozygous individual whose genotype is given, that is capital A, small a, capital B, small b, capital C, small c, what will be the proportion of gametes that carry capital A, small c, and capital C? The genotype of the gamete is given, and you have to find out what proportion of that gamete whose genotype is mentioned in the question. So what is the proportion of that particular gamete once the gametes are formed by the heterozygous individual? So from the question we can understand that the individual is heterozygous, so it will have both the alleles, that is dominant and recessive alleles, and it is a diploid organism, so it will have 2 chromosomes, and the picture of the chromosome might look something like this, that is capital A, capital B, and capital C genes will be on one chromosome, that are the dominant genes, and small a, small b, and small c, that is the recessive genes, will be on the other chromosome. So you have to find out the proportion of this particular gametes, capital A, small b, capital C. This particular gamete you have to find out. So it is, if you see carefully, you will find out that it is a product of a double crossover event. In a double crossover event, two of your genes with respect to one particular genes, they change their position. So what happens? You get a recombinant gamete. So you get a recombinant gamete, and you will get 2 different types of recombinant gametes. So you have to multiply the probabilities of particular gene segments, that is A. The probability is 0.1 into 0.2 because these 2 genes are shifting their positions. So you have to multiply the probability of these 2 genes of shifting their position. That's how I consider them. Even though we say that the 10 map units apart, which means that if we say that AB is 10 map units apart, which means that the chance of AB shuffling its position with respect to B is 10 times in 100 meiosis events. So this is how it means if we see theoretically. So 0.1 into 0.2, which will give 0.2, that is a frequency of double crossover event. And as a result, you get 2 different types of gametes, but you have to find A, B, and C. So we divide it by 2, which will give you 0.01. And the percent, if you want to calculate percent, just multiply the frequency with 1%. So from one more point which highlights in this question is that recombinant gametes are very less in number. The recombination event is very less in number. So if you have 100 gametes, so from 100 gametes, 1 gamete might be recombinant. On to the next question. The next question is, a bacterial culture which has 5 into 10 raised to 8 cells per ml is maintained in a chemostat of working volume 10 liters. If the doubling time of the bacteria is 50 minutes, the required rate of flow of nutrients is dash, and the unit is ml per minute. So first of all, it's very important to understand that chemostat is a type of bioreactor wherein you continuously add nutrients in the feed flow. At the same time, you remove byproducts which are generated in the bioreactor at the same flow rate. So the volume is maintained constant in a chemostat. So and the flow rate is governed by this mathematical equation, that is D, that is dilution rate, is equal to F by V. So F is the flow of nutrients and V is the volume, and that dilution rate is found out to be equal to the specific growth rate mu of the particular bacteria in the chemostat. So this is the one equation that you have to keep in mind. Volume given here is 10 liters, which is 10,000 ml. So I hope everyone understands how to convert liters into ml. The next thing that you have to find out is D, but you know that D is equal to mu. So you have to find out mu. So you can find out mu, that is specific growth rate, from the doubling time. The relation of the doubling time with the specific growth rate is TD, which is the doubling time, is equal to ln of 2 upon mu. So you can find out mu from the equation, which comes out to be 0.0138629 minute inverse. Keep in mind that the unit of mu is time inverse, and the time here is minute, so minute inverse. So from the equation you can find out that is the flow rate of the nutrients is equal to D into V, but D is equal to mu. This is how you have to run your chemostat. Dilution rate should be equal to the specific growth rate, and your dilution rate cannot be equal to mu max, because if your dilution rate is equal to mu max, the washout might occur and there might not be any cells maintained in your bacterial culture. So put the values of mu and V and you'll get 138.6294 ml per minute. That is the flow rate. The unit of flow rate is ml, that is volume, per minute, that is time. Moving on to the fifth question. A stirred tank reactor is scaled down from 10 meter cube to 0.1 meter cube. Large tank dimensions are D, which is equal to 2 meter. And the dimensions for the small tank are D, which is diameter, which is equal to 0.5 meter. And the RPM, that is revolution per minute, is 100. On scale down, you have to scale it down and you have to make sure that it has a constant impeller Reynolds number. So what is the rotational speed required for the impeller of the small tank? So you have been given two quantities, that is diameters of both the tanks and rotational speed of the smaller tank, rotational speed of the larger tank, and you have to find the rotational speed of the smaller tank. But you have to make sure that your impeller Reynolds number is same. So let's not be confused between Reynolds number and impeller Reynolds number. These two are different quantities. So impeller Reynolds number formula is N, which is the rotational speed, into D square, that is diameter square, into density upon viscosity. So I think viscosity will be, so this is general formula for impeller Reynolds number. So from this we can derive equation. So your viscosity is going to stay same because you are going to use the same medium in both the tanks, so viscosity is going to be the same. Density is also going to be the same. What is changing is your rotational speed and diameter. So nd square, the nd square, that is rotational speed into diameter square, this quantity should be same. So if you're decreasing one quantity, the other quantity increases. If you're increasing other quantity, the second quantity which is related to it decreases. So this you have to keep in mind. So nd square 1 is equal to nd square 2. So put the values for larger tank, it is 100 rpm into diameter is 2. So 2 raised to 2 is equal to n2, which is rotational speed for the smaller tank, into 0.5 square. So after solving this equation, you will get rotational speed for the smaller tank, rotational speed of the impeller of the smaller tank, which is equal to 1600 rpm, revolutions per minute. 3 micrograms of circular plasmid of 4200 base pair was digested with the restriction enzyme and subjected to agarose gel electrophoresis. 5 DNA fragments of different sizes were observed, and the sizes summed up to 4200 base pair, which is understandable because plasmid is 4200 base pair. So of course the summation of the different molecular weight bands will be 4200. The number of picomoles of DNA ends generated after complete digestion of the enzyme is. So there is 3 micrograms of circular plasmid you have, and you have digested it with one particular enzyme, and that enzyme gives 5 different fragments of varied lengths. And from this you have to find out how many picomoles of DNA ends are generated. So if you're considering one fragment of DNA, it has two ends. Similarly, 5 fragments of the DNA will have 10 ends. So we will go with the straightforward formula for moles. So consider if your circular plasmid is not digested, so how many moles of circular plasmid you will have? So moles of any compound, so here it is plasmid, so moles of circular plasmid will be grams of the plasmid divided by its molecular weight. So there is 3 into 10 raised to minus 6 grams divided by 4200, because there are 4200 base pairs, and molecular weight of single base pair is 650. So this is the figure that you have to remember all the time. It is a constant, 650 for base pair. So 4200 into 650, you will get the molecular weight of the given plasmid. So your answer for the moles of circular plasmid is 1.098911 into 10 raised to minus 12 moles. So this is for one plasmid. This is for one particular plasmid. But this plasmid is divided into 5 fragments, and 5 fragments have 2 ends. So there are total 10 ends. So moles of the end will be 1.098911 into 10 raised to minus 12 into 10, which will give you 10 picomoles. So pico prefix here stands for 10 into minus 11. Sorry, it's not 10 into minus 12, it's 10 into minus. The average DNA content of a single microbial cell is 4 femtogram. A soil sample analyzed for its microbial community DNA is found to contain 0.32 microgram DNA per gram of the soil. The number of microbial cells per mg of the soil are. So first of all, you have to know that the 4 femtogram means 4 into 10 raised to minus 15 gram. So there are different prefixes that you should know, that like micro, picomoles, pico, or femto, kilo. So these are the prefixes that are used most of the time. So you should know what femto stands for. So here femto is 10 raised to minus 15. So DNA content per cell is 4 into 10 raised to minus 15 gram. And you have per gram of the soil DNA content, that is, there is 0.32 micrograms of DNA content per gram of the soil. So it is straightforward that number of cells per gram of the soils will be the total gram of the DNA divided by DNA content in one single cell. So once you divide 0.32 into 10 raised to minus 16 grams of DNA content which is present in the soil divided by the DNA content of the cell, which is 4 into 10 raised to minus 15, which will give you 0.08 into 10 raised to 9 grams, 10 raised to 9 cells per gram of the soil. Now after conversion, you get 8 into 10 raised to 4 cells per mg. So it is very important that you convert decimals into whole numbers appropriately, because most of the times it might happen that you may make mistake while converting. In spite of arriving at the right answer, you might get your answer converted in wrongly. So it is very important that you convert the answer appropriately. So number of the cells per mg of the soil is 8 into 10 raised to 4 cells per mg of the soil. Ninth question. You have induced a rare mutation in a microbe which is in special media, has 50% higher specific growth rate. That is 2/3 of the doubling time for the normal cells. If the mutation frequency is 1 in 10 raised to 6 cells, how many generations of normal culture are needed for the population of the mutant and normal cells to be equal? So here you have to imagine that there is one cell which is a normal cell. It divides and produces a cell population of 10 raised to 6. So from that 10 raised to 6 cells, one cell will be mutant. And from that point onwards, if the cell starts dividing, and from that generation onwards, if the cell starts dividing, there will be a certain point in time where the number of mutant cells will be equal of normal cells. So here they have given that it has 50% higher specific growth rate. So it has 1.5 times higher specific growth rate than the normal cells, and doubling time will be less because it is multiplying faster. The mutant cells are multiplying faster, so it will have doubling time less, that is 2/3 of the normal cells. So suppose normal cells are 2 raised to n into 10 raised to 6. So 10 raised to 6 cells, from 10 raised to 6 cells, after nth generation, the number of the normal cells will be 2 raised to n into 10 raised to 6. And from that one mutant cell, from that point onwards, the mutant cells after a generation will have number of mutant cells is equal to number of normal cells. So mutant cells will be 2 raised to a. So keep that in mind. You have 10 raised to 6 cells, normal cells, and 1 mutant cells. So after nth generation of the normal cells, your normal cells will be equal to mutant cells, and after a number of generations of mutant cells, your mutant cells will be equal to normal cells. So n and a are number of generations. So from that point onwards, after time t, both mutant population and normal cells will be equal. That is, m is equal to n. So n, which is equal to t divided by the doubling time for normal cells, t divided by td1, and a, which is equal to the total time required for reaching the a generation, that is t divided by td2. So, but actually your td2 is lesser by 2/3 than td1. So td2 is equal to 2 by 3 of td1. So put these values in a and make your variables same. So after substituting the values, that is, we get 2 raised to 1.5 times of n is equal to 2 raised to n into 10 raised to 6. So after solving, you will get n is equal to 39.86, which is approximately 40. So this question has asked you to find the nth generation of the normal cells. After that nth generation, your both normal cells and mutant cells will be equal. A closed circular bDNA of 1050 base pair has 94 turns of helix, opposed to the expected 100 turns. A superhelical density of this DNA molecule will be, so we have to calculate superhelical density. Before that, you have to know that linking number, which is equal to the twist plus turns. The twist is one DNA strand. How many times one DNA strand twists around another DNA strand, plus the number of turns that particular DNA takes around the helix. The sum of these two quantities will give you linking number. But superhelical density is is the ratio of number of turns removed upon number of original turns. So there are 10 bases per turn, 10 bases per turn. So now there are 94 turns, but the expected turns are 100. So 94 minus 100, which is 6 turns. 6 turns are less. So minus 6 on the number of original turns. Number of original turns are 100. So 94 minus 100 upon 100 will give you minus 0.06. So this is the superhelical density. And if the number of turns are less, then your DNA molecule is coiled, is either positively coiled or negatively coiled. So the answer is minus 0.06. That's all for today, guys, and thank you for listening and watching the video. Till then, keep questioning. Goodbye.